This is the video notes for Chapter 10, Section 1. And just to give you an idea of where we're going, Chapter 10 focuses on cell growth and cell division, including mitosis. And then we're going to move into Chapter 11, which is meiosis. So this is biology with Miss Mannion. So why are cells so small? If you think about the cells that we've looked at, they've always required us to use a microscope. If you look down at your hands, it's really impossible to see the individual skin cells. So why is that? Why don't cells grow and grow and grow and continue to grow until they're very large? Well, there's a couple of different factors that influence why cells don't grow as much as we might expect them to. There's two issues related to cell growth. The first one is the larger a cell becomes, the more demands the cell places on its DNA. The second one is the cell also has more trouble moving nutrients and waste to and across its membrane. So these are the two main factors that are influencing why cells don't grow and grow and grow. So the key point here is the smaller the cell size, the easier it is to do what the cell needs to do. So let's go on and investigate each of these. So first one, DNA. The issue with DNA is in small cells, the amount of DNA is sufficient. If cells grow any too large, their amount of DNA is not going to increase. So they're kind of stuck with the DNA that they originally had. So that's not going to grow along with the cell. The problem with that is if not enough DNA to go around for all proteins to be made and other cell processes to continue. So basically the cell runs out of DNA. So that's a big thing, and that's why cells don't continue to grow bigger and bigger, is because this lack of DNA to help synthesize proteins and help daily cell processes to continue. The second one is the exchanging of materials. So if a cell size is too big, it requires too much energy to be used to transport materials throughout the cell and to bring in or remove things from the cell. So now we're starting to talk about active transport and the cell having to use energy in order to get things in and out of the cell. The cell wants to be as efficient as possible, so at a larger size, this is going to be a lot harder to do. Cells also rely on diffusion, so things moving from areas of high concentration to low concentration, and diffusion over large distances just takes too long to be efficient. So therefore, the cell is going to want to remain somewhat small in order to make these processes more efficient. The key here is the relationship between a cell's surface area to volume, and that's what we're going to look at next. So if we look at the surface area and the volume, cells with the largest surface area to, that's interesting. Not sure why that's picking up here. All right, sorry about that. Surface area to volume. Cells with the largest surface area to volume ratio are the most efficient. It is much easier for small cells to perform the normal functions than a larger cell. Smaller cells are more likely to survive and divide than larger ones. And the smaller the ratio, the harder it is to get waste out and could cause cell death. So when we think about this, it's just a lot better for cells. If they're smaller, they're able to use their DNA effectively and transport things in and out of the cells. So you want cells that have a larger ratio, so generally more surface area to volume. And what happens as the cell gets bigger, that ratio decreases. So that's what we're going to kind of look at for this one. So this is a surface area to volume ratio. So when we look at the first one, I'll see if I can turn my little clicker on here and actually do something for this. All right, so we're looking at our first one here. Okay, you have your surface area. You know, this is a cube, so you're going to find all of your surface area. Height times width times number of sides, number of boxes. So what happens was, and in something smaller like this, you get a volume of 1 and our surface area of 6. So you get a surface to volume ratio of 6. But what happens as this gets larger, so we notice in this one here that our box is getting larger, our ratio is decreasing. And so think about, the nucleus inside, or maybe your big old vacuole inside. As the volume increases and the surface area doesn't increase as much, it's going to be a lot harder for these materials to get in and out of the cell and for the DNA to work efficiently. So this just gives a little bit of visual of how kind of that relationship happens, but the main thing is you want a larger, wow, I'm really bad at this. Sorry guys, this is my first time using Screencast-O-Matic, new thing. Larger ratio, that is the key for here, is that you want that larger ratio because it makes the cell more efficient. So the last kind of piece of this is we talked about why cells don't get bigger and bigger and bigger. So how do we as organisms grow? Like, how did we come from this little fetus inside our mom's stomach to where we are now, and why do you continue to grow? That has to do with the division of the cell. So if we look back here on our chart here, we notice that this final version over off the right here actually has the same total volume. However, because there's just a bunch of these smaller cubes put together, we have a larger surface area, and that increases our surface to volume ratio. And that's where we want it. The higher, the better. So because of that, that's why we do cell division. So you can have a bunch of different little cells still adding up to the overall volume, but also increasing the surface area. So that's why cell division occurs. And cell division is when one cell divides into two new daughter cells. And because there's this division, genetic material is going to be replicated before the cell can divide. That way, each cell is going to have DNA. And cell division reduces the cell volume. This allows efficient exchange of the materials with its environment, and also make sure that the DNA can work properly. So this is what we'll go into in the next section. So this ends 10-2 notes.